Hello, everyone. This is a presentation that was just given at Northeastern University for a CPDA conference, and it covers microbial environments on cannabis and the impact these microbial environments have on safety testing and the impact these things may have on healthy grows. It covers a broad area, but what we're really going to zoom in on are the regulatory implications of running microbial testing with the current systems that have been used for probably too long. These are known as culture-based systems or petri dish systems, versus how we could leverage the power of genomics to change this game for the betterment of both yield for growers and safety for patients. It's actually a win-win when you do this right. So let's dive into it, okay? So there are two papers that most of this presentation is based on. They're open source. I encourage everyone to go and see them. They're at F1000. F1000 runs an open review process, so you can see the names of the reviewers on these papers are like members of the community, like Ethan Russo and John McPartlin and Justin Vishetic, a variety of very well peer-reviewed, published people in the field. And you can read the reviews. They're very thorough, actually. Whenever they're open reviews, you'll see there's people put a lot of care into the review because their name stays on it publicly forever. So we have sequenced now over 300 microbiomes, and I want to explain what that means momentarily. But the microbiome is really the DNA from all of the microbes that are on the plant. In addition to this, we have also done a fair amount of comparison to traditional systems that would measure microbes. These are really culture-based systems. And culture-based systems, there's an interesting dilemma with culture-based systems. Most of the industry standards are set according to how these systems work, but we've now discovered with the power of genomics that the vast majority of microbes you can't even culture, and so most of these things are blind to the vast majority of what's going on this plant. And so that's something that just needs to be kept in mind as we go through this data. Okay, so there are 6 tests we're really going to focus on that you'll see listed in the Pharmacopoeia. This is the monograph for Cannabis, and you'll see these tests are generally adopted in many states, although you'll see California and Florida are wisely deviating from these. But they're effectively what's known as a total aerobic count test, a total yeast and mold test, coliform, enterobacteria, E. coli, and Salmonella. And these tests are done with a variety of technologies, but most of them done today are with culture-based methods, and we're going to show you a new way to do these. And it's called ITS-PCR in sequencing. ITS stands for an internal transcribe spacer. These are conserved regions in all living things, okay? With the exception of viruses, everything on the tree of life has an ITS sequence. Now, depending on whether it's fungal or bacterial, they'll have different names for these, but fungal ones are usually have 18S ITS regions, and bacteria tend to have 16S regions. And so if we want to amplify DNA that we know is from all fungal origin, we can do that by designing primers that hit the 18S region and will amplify everything that's deemed to be a fungus. If we want to do this with bacteria, we target particular sequences in the 16S region, and we can amplify those and only amplify bacteria. And so this can be done to effectively replace the total yeast and mold growth plates that are out there and the total aerobic count plates by targeting these at conserved regions in the DNA of many organisms. Now, while the primer sites sit on conserved regions, the regions they amplify here in black, you can see in this graph, are actually hypervariable and very specific to each organism. And so these are really the molecular barcodes that are used for the whole tree of life taxonomy system now. No longer are people looking at organisms and deciding what they are. They're using DNA as the proper catalog. So this should be really the centerpiece of food testing in our mind, and likewise cannabis testing. Now, what you do when you have PCR primers that can amplify a particular genre or particular species and genuses, what you can then do is quantitate how much of that entire genus is present. And we do this with a system that we sell called Pathogen Indicator. Okay, so all conflicts in the table. This is a presentation. about many of the tools that we use and that we sell. But hopefully we'll convince you that this is in fact the right way to do it. So quantitative PCR turns that amplification rate into a signal so you can get an understanding of the amount of DNA that's present. And when we do enough sequencing in this arena, where we take those PCR products and then sequence them, we begin sequencing across those conserved primers that amplify the entire genre, but the whole entire genus, I should say. But also, we capture the internal region, which is a tight signature for the actual species that's there. Now, we don't always get perfect species resolution with the method that we're using here. This ITS sequencing does get down to the genus level with high accuracy. Species level is sometimes needs confirmation. But nevertheless, we can sequence these PCR products if we want to get full microbiome IDs. And when you do something like this, you get a very interesting distribution. Here are just a select set of samples that we have done this on. And you can see there's a very long tail of different organisms that are present on this Girl Scout cookie, and you can see some penicillium species in there that we have highlighted in red. And this paper in particular focused on a few of these penicillium species because they had not been cataloged in cannabis before, and we had found that they have the potential to make some very interesting compounds that everyone should be aware of in the extraction process. But you can also see other samples that had less diverse microbiomes, but they still have some aspergillus. And what you're reading on these bar charts is the number of reads that we found that match that organism's ITS sequence. Okay, so you're seeing these numbers, 77,000 and 50,000. These are, I'm sorry, those are hundreds of thousands, if I'm correct in some cases, but large read numbers. So it's with the sequencing that we can really begin to itemize what is present on that plant, independent of a filter of what can potentially grow. When you're just viewing the microorganisms that can actually culture coming off the plant, you're really only looking at like 5% of the diversity that's there, if that. And what you're going to find is that this is actually extraordinarily important when it comes to cannabis. So this paper went and demonstrated that, hey, look, there's a lot of things that don't culture on multiple different platforms. 3M didn't pick any of this stuff up across their total coliform, their total entero, their total aerobic, or their total yeast and mold. Likewise, Biolumix, likewise, Simplate, all blank, yet we can see something. This is a reminder that 95% of microbes don't culture. Now, a lot of people say, hey, we don't want this, you're going to fail too much stuff. You're picking up what could be dead stuff or stuff that doesn't culture. Well, what cultures on Petri film is not really relevant. It's what cultures in a human lung that's relevant. The Petri dishes are actually no surrogate for that. And while we can certainly pick things up, we can also set thresholds for when the amount of DNA that we do see is in fact a threat based on how much of it is there and how much of it is viable. So we can discern whether something's viable or not with PCR, and we're going to touch on that as we go forward. But that's an important detail to keep track of, and it's also important to keep an eye in these papers in terms of the absolute fail rate. What you won't see is that PCR is having a higher fail rate next to culture, and that's because we don't pick up a lot of other confusing artifacts. One thing that we noticed in all these culture-based systems is that they're not very specific for the bugs that they claim to grow, like the total yeast and mold plates grow bacteria. And when that's the case, you fail things for beneficial microbes like bacillus. So while PCR in this one case we're showing is picking something up that doesn't grow, we'll show you many other cases where the films are picking up things they should never they shouldn't be triggered by at all. So we'll touch on that momentarily. So the long end story of this paper was that we found different microbes no one's seen before by using DNA-based methods, and we found that they could potentially make mycotoxins that could harm patients. And these mycotoxins, if you look at them, they have peer-reviewed publications on how they may interact with the cannabinoid pathway, which could be problematic. And it appears as if they're lipophilic and nanomolar. That means they could be 100 to 1,000 times more potent than CBD, and they may enrich more favorably in various extraction methods. And so, just like pesticides, we've got to be very careful whenever we see nanomolar mycotoxins that could be present. Now, to be clear, we have not detected these as molecules. We've detected the genes, and we're going to go into that. Before we do so, this is just another paper from Duncan Ryan that highlights how citrinum, which is another mycotoxin, a potential mycotoxin we found in cannabis. It was actually the microbe that makes it we found in cannabis. We didn't actually find the toxin. That's a very key difference. But you can see that if that toxin is getting produced, it is in fact potentially a target for interfering with cannabidiol, just like Paxilene. So this is what these two molecules look like. And you can we had to go and do confirmatory PCR and sequencing to get this through peer review because there's always a concern that the ITS sequencing didn't get very specific down to the species level. Well, we can see the genes that make these particular toxins are in fact present, and they sequence very well. We have begun on a fairly arduous process of figuring out how to detect these mycotoxins in cannabis, but that's not yet a completed story. There's a lot more work to be done there. We've been trading some notes with NIDA on how to best do that, and at the moment we're replicating other people's work with LCMS to pick these compounds up. So let's not raise any alarm bells here. We're not seeing the compounds, and that's very important for people to know. Evidence of the microbe in the gene is not evidence that it actually expresses it in this environment. So let's not get too worried about this until the actual presence of the compound can be firm. But it is a reminder that these are things in the plant no one's looking for. So let's see what they are looking for. Well, if you look at some of SC Labs data, they presented this at CanMed in 2016. These are the typical fail rates that they see. It's close to 15% are failing for microbials, and the reason all these numbers don't add up is because some samples fail for both things, and so the yeast and mold, even though being 14.3 and E. coli 4% and coliforms 1.7, this is not all summing up to the 20% you'd expect. It's because many samples fail for two things. Now you'll notice the thing they fail the most for is for yeast and mold, and when there is a high failure rate for yeast and mold, what it tends to do is induce people to use lots of fungicides. And so when you begin to look at the SC data on the frequency of the pesticides that are being used, they're mostly fungicides. All right, so if we have tests that aren't doing a very good job of measuring yeast and mold, we're probably going to have excessive use of fungicides like Microbial, which are not currently regulated as stringently as any of the microbes. And so this could be a positive feedback loop we got to keep our eye on. If these yeast and mold tests aren't correct and they're giving out false positives, well, then they're doing more harm to the community than good if it's inducing a lot of microbial use. So, okay, so let's move on to the next study, keeping those things in mind. Some of the questions that came out of this first study that we tried to address in the second was, do the microbials match the original microbials coming off the plant and then the ones that you see coming off the culture systems after their period of growth? So this is just really a question of what we're actually measuring on Petri film. If you put material from a plant onto a Petri film or an agar plate and grow it for 48 hours, do you get the same ratio of DNA that's going on the plate versus what's coming off the plate afterwards? Very important question. And what percent of the CFUs per gram are actually what they expect them to be? So if you're on a total yeast and mold plate, are you getting colony-forming units that are yeast and mold, or are you getting bacteria? That's a question that's not really been addressed yet to date with many of these technologies, despite them having been around 100 years. So colony-forming units is what CFU per gram is, and we're going to dive into what this all means. So what you're going to find, the executive summary here, is that qPCR provides very specific microbial risks in under 24 hours. Most of these culture-based systems are failing too much material, and they're failing to grow the important risks. So they're getting triggered by what we would call beneficial microbes, and they are not growing the actual harmful ones for patients. So they're doing exactly the opposite of what we want them to do, yet they're still the most commonly used thing in cannabis safety testing. So just heads up. So we'll touch on why in a moment. But ideally, you want culture systems to allow beneficial microbes to go through because that can lower your fungicide use. You don't want to penalize people for using those. And at the real whole point of doing these microbial testing is to actually pick up Aspergillus and the human harms, and they're not doing that either. And so you have the worst of both worlds when you think about using culture. Okay, so why? Well, there's a lot of false positive total yeast and mold tests, as we mentioned, which we believe are encouraging improper fungicide use. Where you're going to see in this study where over 60% of the DNA that we get off of a total yeast and mold plate from 3M is actually bacterial DNA. Same thing's true with the culture-based systems from bioMérieux. We are getting basically other organisms growing in these things contributing to the readout, and that's a big problem. The other thing we're able to do with DNA is we're able to find things that are anaerobes, things that may not grow in an aerobic culture system, but may in fact grow in an anaerobic oil. Clostridium botulinum is something everyone in the food industry knows about, and they are petrified of the implications of this, making Botox and oils. If this bug survives, the spores survive, and our DNA-based sequencing implies there's DNA and there were spores present in some of these extracts. This implies we could be finding botulinum outbreaks in cannabis material in the future if we're not careful. So again, aerobic-based culture systems can't do this. So this was also published, peer-reviewed in F1000, and I think one of the most telling graphs in this entire study is the one that everyone asks us, is do the CFUs per gram match the CQs? And the answer is, for all the species that in fact do grow, we can nail the CFU per gram to a CQ. And you can see that with all these different Candida species here that we know how to culture. When you get to things like Aspergillus, which don't culture, we can see it with the DNA, and it can't be seen with the culture systems. And there's a reason for this that was described by Becker et al., and it's something known as heterogeneous macrocolonies, which means Aspergillus, when it gets into solution, has this capacity to form a hyphae and a network and clump, and all of their CFUs, if you will, colony-forming units, begin to ball up into one big chunk. And if you happen to miss that big collapsed clump, if you will, you're going to get nothing to plate. Whenever you get clumping in scenarios like this, your CFU per gram end up suffering from a sampling problem. When you go in to sample the solution, it's really clumpy, and there's thousands of cells all clumped together, and you happen to pull out 1 ml and miss that big clump. You don't have them uniformly distributed into solution, and so the liquid handling there is not representative of the true CFUs per gram. And so this causes what you're seeing in that previous slide, a threefold or 3 log scale differentiation between qPCR and the unculturable Aspergillus. Now they do culture, they just don't culture quantitatively. And so they introduce massive sampling biases when you try to culture these things in liquid culture. When you use PCR, we're getting a more accurate representation of the actual threat level. And this is important because Aspergillus is the only thing that's ever killed anybody in cannabis. So we've got to get this one right. All right. So when you go and try and plate these things, what you tend to see are these hamburgers that end up on a lawn. They don't look like a tiny colony. They almost look like a huge mushroom. And that's because those are all being seeded by 1,000 cells at once, and they take off. And it makes it very difficult for you to really do serial dilutions and be real certain whether or not your lab tech has properly sampled the solution to find an Aspergillus risk. Whereas with qPCR, we end up melting all these spores away and just measuring DNA levels, and we can get very precise on the number of copies that are there, and we won't have a sampling bias with a 1 CFU per gram type of threshold test. So important to do all these things on ATCC stocks. We want to underscore that. To do these appropriately, most labs should be operating with a BL2 laboratory so that you can spike human pathogens onto samples. Hopefully you can use attenuated ones to get the job done. But this is really required to validate any test, is you have to spike these samples in, these bugs into your matrix to see if your matrix is actually an antibiotic, much like cannabinoids and terpenes. It does affect the results, and that validation is really matrix dependent. So the next thing that was very critical to this whole study was to look at this Heisenberg uncertainty effect. What does the DNA look like coming fresh from the flower? What does it look like after you use a measuring device, much like a culture system, which is really another evolutionary experiment, if you will. It's putting bugs on a new carbon matrix and seeing which ones survive on the new environment and which ones die because you've changed the environment from cannabis to agar. Well, So we looked at DNA both before and after plating on 2 different culture systems, not to pick on 3M. We also looked at a culture-based system that was more liquid format with Microbiome. And in doing that, we could qPCR and sequence the DNA both before and after and get correlations as to how well our CQs matched up with what was viable and what was the species breakdown. And the most interesting thing that we saw with the species breakdown here was that before and after sequencing profiles were completely different. Okay, so this is really important to sink in. This bar chart here, everything in gray is actually the sample before it went onto 2 different culture systems. And then everything in orange and blue is what it turned into on the various culture systems afterwards. And so you can see there's almost zero correlation with before and after, and each platform has its own bias. The sample after Biomareu has a tremendous amount. I think that's a Pseudomonas species in there, and maybe another Pseudomonas species you can see all the way over to the right. And when we start looking at the 3M platform, we see a completely different spectrum of Rostonians. So this is shocking. It means the act of measuring the microbes off of cannabis has absolutely no correlation with what was initially on there. You're just presenting it with a new environmental stress, and a different evolutionary set of microbes persists and grows and gives you a tarot card of concern. You can also see this in a few of the other charts here where you look at before and after, and you can see quite a striking change in the before and after charts here. In particular, the gray to the blue is quite striking. Now, one thing that you can also do with this data is you can look at what's known as beneficial antagonism. This is when you see certain microbes present that knock out other harmful ones. And so these is what people in the industry may know as beneficial microbes, like trichoderma and bacillus. Bacillus isn't on here as a bacteria, but we tend to see whenever there is trichoderma present, Penicillium and Aspergillus don't seem to survive in the aftermath. So you can see in the lower right here, and the graph C, we have Penicillium and Aspergillus present in the before fraction, but since there was some trichoderma present in the before, it took off in the after and wiped out the Penicillium and Aspergillus, and that's exactly what beneficial microbes are supposed to do. So keep using those, except ask your testing lab if you're going to be tested on a total yeast and mold plate, because these will grow these will grow more favorably. You will actually be penalized for using trichoderma on a traditional total yeast and mold readout. And the other graphs you're seeing here are what happens in the Biomareu platforms. We have those all documented with nice photographic images, and you can see there is a complex assortment of different microbes there, just based on the color you're seeing in some of these growth chambers and some of these Petri films. But that is actually a representation of an evolutionary experiment that you perform by putting those microbes off of cannabis, which is a terpene-rich environment, and communication with microbes onto something that is not expressing terpenes and cannabinoids, and seeing which microbes suddenly show themselves afterwards. This is not the same environment as a human lung, and so that's a really important thing to keep in mind. So this obviously raises a lot of Heisenberg uncertainty problems for regulators, because the act of actually measuring the microbial load changes the load in front of your eyes. So it also raises the question, well, do the 2 different culture systems agree with one another? And from a sequencing standpoint, the answer is no. We have very little agreement between the total counts that we're getting off of a 3M system versus a Biomareu system. So here's an example of looking at the before and after total yeast and mold counts. And this is giving you another picture from the bacterial standpoint that you're actually getting a lot of stuff down here on the horizontal axes. You're seeing this was almost had no presence before, but afterwards all of these bacteria popped out. Basically, this is 16S. So we're measuring bacterial DNA on a total yeast and mold cartridge. So bacillus is on there. Bacillus, we know, is often used as a beneficial microbe. So if you use that, it's going to grow fantastically on a total yeast and mold plate, and who knows what it does to CFU's count, but we know its DNA is quite prevalent. So what you're seeing on this x-axis are the organisms that were not at really high levels before, but came to very high levels afterwards on a total yeast and mold plate. And when you see bacteria on there, you know those total yeast and mold plates are having a heyday with bacteria. When you look at this on the 18S side, so this is before and after culture for total yeast and mold plates, and looking at the what amplified with 18S sequence, so what type of fungal background do we see? And what we can see are various Penicillium citrinums that are showing up, so that one's still on our radar. But these are more or less showing up in a diagonal ratio as you'd expect. And just keep note on the axes here, they're different scales. And so they're certainly viable because there's a lot more of them in magnitude after, in terms of DNA, number of DNA reads, than there are before. And so these are viable just based on the slope of the curve on those axes. And then we again look at that question about correlation. Well, here are the 16S counts, the bacterial DNA sequencing numbers that we see off of Microbiome in the after portion on the y-axis there, and the 16S counts we see in 3M in the aftergrowth on the total yeast and mold plates. Okay, so this is bacteria. How do the bacteria differentially grow on these different culturing systems when they're supposed to be culturing total yeast and mold? They both culture bacteria, and they both do it differentially. And you can see the things that are on the x and y axes as straight lines, those are things that uniquely grow on one platform or the other. There might be an antibiotic we've heard in the Microbiome platform of chloramphenicol, which would probably do something like that. And then you can also see the things right in the middle that may have a little bit more of a slope and more of a correlation. In total, though, the correlation is horrible. It's 0.196. When we move to the total yeast and mold side of this equation and ask how many things in this lower right graph, how many things are on the 18S qPCR and sequencing for total yeast and mold in the Microbiome platform, that's on the up axis, and on the horizontal axis, we are asking the number of fungal DNA sequences we see, 18S, on the total yeast and mold Petri films, and the correlation is much tighter there. It's a 0.992. So they agree on the fungus they do grow on those plates, but they differentially are contaminated by bacteria. And I just want to remind everyone that 60% of the DNA coming off of these things, 60% of the reads in total were 16S reads. So there is a substantial amount of bacterial growth on these plates. Now, one thing you can do with sequencing is it's digital. You can then count numbers. Like, what is the 18S specificity? In other words, when I amplify with 18S primers, which should target fungi, how many times do I get bacteria DNA coming through? Because that tells you how specific your actual test is. We just saw that 60% of the DNA on total yeast and mold culture systems is bacteria. Can we beat that with specificity of primers and DNA? And the answer here is unequivocally yes, by orders of magnitude more specificity. So when we do this, we see maybe a couple hundred hits, reads that are 18S primers that might map to bacteria. However, hundreds of thousands of these reads actually map to the proper targets. They map to fungi. So there's very high specificity for the 18S primer sets. We look at the 16S primer sets. It gets a little bit murkier because 16S primers there's a certain feature of those, in that they happen to also be in fungal mitochondria. If you remember, fungi have had a genome acquisition of an archaea or a bacterial organism a very, very long time ago that created mitochondria. And so the mitochondria have a little bit of bacterial signature in them. And so what we tend to see are 16S primers, again, having very high read numbers that are all bacterial, but a little bit more noise when it comes to finding some 16S sequence with these 18S hits. And that, we believe, is due to this this fungal mitochondrial contamination that occurs, and that there's probably not much we can do about that other than better classify reads that appear to be mitochondrial. And we may be able to reduce some of that number. But even so, these are far better numbers from specificity, and we have now a database of everything our primers actually hit. So we can tell you with high confidence whether we've ever seen something before, and whether blast searches would pull back anything else we have never seen before. This is not something, you cannot do this with culture-based systems. There's not enough samples in ATCC to test this level of diversity of organisms, and it's just really just not doable with the old paradigm. And so this brings us back to this positive feedback loop. If we have a microbial testing system that cannot discern between beneficial and harmful microbes, where both of them grow on a plate, and we're handing back failure rates, which could be due to really good organic practices of using trichoderma, we could be signaling to growers to use more fungicides, which is exactly the opposite of what we want to have happen. Okay, we really need to have higher specificity that targets the harmful microbes and doesn't penalize people for using beneficial ones, because it's the beneficial microbes, as we've shown in some of these slides, are actually the key to reducing the harmful ones. This beneficial antagonism has been published; it's real. So keep your eye on it. So this slide just summarizes a few of those points. We want to reduce pesticide usage. We have to get more specific microbial testing. Beneficial microbes are known to reduce the need for pesticides via beneficial antagonism, and beneficial microbes really shouldn't be included in any CFU per gram count because they're not a health hazard. And when we start thinking about techniques for this, we should keep our eyes aware of where the FDA is going. They are building a genome tracker network that does DNA sequencing and performs that sequencing and to catalog outbreaks. So when you get in the world of foodborne outbreaks, no one's using these Petri films. They're all moving to DNA sequencing to get rapid turnaround time and precision in actually what they're measuring. No more guessing what a colony is. That's not real science. That's just reading tarot cards. Now, speaking of real science, there is a lot of science involved in trying to get marijuana-infused products to culture on these Petri films, because we're finding that many of these marijuana-infused products have compounds in them that inhibit the indicator and the chemistry of a lot of these Petri films. So many of these Petri films have some type of indicator that gives you that dot when a colony forms. And these are experiments we've done just to take simple MIPS and dissolve them and put them on 3M and see whether or not they inhibit growth based on having a known amount of spiked in E. coli at the same concentration. And what you're seeing in these graphs are 24 and 48 hour images of what these things look like. And in the upper left here is a tack plate that has E. coli as a blank. And then we have a spike in of a known amount of E. coli into TSB. And we can then measure that in comparison to what happens if any of the candy is present. And you can see the candy is just shutting down growth. Now, the real question is it shutting down growth, or is it killing the indicator? We have some evidence via qPCR it's actually killing the indicator. There's actually CQs that are pretty close between these two things, but the actual representation on these 3M plates is one that implies the candies interfering with the readout mechanism. And you can see this with again, at 72 hours and 96 hours, you begin to get some recovery of these things, so perhaps a slower growth environment. So here we have, again, the same thing done on different forms of plates. These are plates that are coliform plates, and interestingly enough, a similar effect happens. We see E. coli alone versus E. coli plus candy, and there's a lower CFU count when the candy is present. This candy was a neutral candy, no cannabinoids. All right, this is just the sugars, not the antimicrobial cannabinoids having this effect, which is a really important question and an important detail. And so you can again see this at 72 hours and 96, that the candies are playing a role in altering the way these things work. So there's really a lot more validation that's required to run these types of Petri films in a cannabis environment with all the complexity of the MIPS that are going on out there. Each matrix really does need to be looked at very closely. We don't have as much of that trouble with qPCR because each matrix we approach, we pretty much annihilate and scrub all the DNA out of it, so that we're always testing a DNA environment without any contaminants there. And while that DNA prep feels like it's more work, what it's really doing is eliminating all of the impurities that are causing these types of validation griefs with Petri films. You can go in circles trying to validate all of these things because there's so many variables. And DNA purification removes a tremendous amount of those variables by just scrubbing the DNA out and measuring only that, getting it away from all the inhibitors, and doing a really clean readout. Okay, so I think there's other websites that go into the work that I presented at CPDA on point of go detection, but we are building, very briefly, we are building these tools that are colorimetric, that just change color if there's a DNA target there. This will be important for all of the presence absence testing that is happening in the field. And some states are moving in that direction for Aspergillus and E. coli. And so these things could readily be adapted for that. We have them running for picking up the CBD allele at the male versus female problem on XY. We're building them for a variety of the cannabis pests that are out there, from powdery mildew, russet mites, and botrytis. You can learn more about that at medicinalgenomics.com under the UPCR section. It's not only pretty to look at, but there are some really novel things that Medicinal Genomics has done here. We are the first people to ever sequence the powdery mildew genome on cannabis. It's unique to cannabis, and it needs its own assay. We've developed that. We've also sequenced the russet mite genome and targeted assays for this. These are all important because one of them gets vascularized into the plant and can spread through cloning, and the other one lays eggs that can spread through cloning. And we can pick up these vascularized mycelium networks for powdery mildew and botrytis long before they've actually sprouted and it can spread. So there is actually an eradication, detection and eradication program that one can do. So with that, in summary, culture-based safety testing really is just safety theater. It has nothing to do with real and health concerns, and we will reiterate once again, there are patients that are succumbing to aspergillosis. It has a 50% fatality rate. It hits the immunocompromised. 2 patients that are immunocompromised in California succumbed to some type of microbial infection in the lungs in 2016. And there was a publication in 2015 from Remington et al. Showing a patient having aspergillus in their lungs, giving them a pneumothorax, and also having the identical DNA in their vaporizer, making the connection that it actually came from the vaporizer. So we don't believe these vaporizers are killing those spores, and qPCR can pick this stuff up, whereas the culture-based systems are going to miss it by 3 orders of magnitude. So we really need to get qPCR in place if we want to have detection for the things that can hurt patients. UPCR will help bring this further into the field. It's more portable, it's easier to use, more friendly to people who perhaps aren't used to quantitative PCR and fluorescence, and anything that's plus minus, it should be able to handle. Quantitative, it's not quite there next to our quantitative kits. It cuts that corner, but it can pick up a lot of the binary things like SNPs and things that govern CBD, THC, and CBD genetics. Y chromosomes, vascularized powdery mildew, russet mite eggs, botrytis, and a host of microbial risks. So, all right, with that, there's a lot of people to thank here, and we will leave it at that. Thank you.